Okay, we're going to go ahead and get started here. So I know a few people will be trickling in. I'm just the warm-up act for Victor, who's going to do the real introduction here. So for those of you who are from outside of NCSA or haven't been to our colloquium series before, we have this colloquium every other week and occasionally in between at NCSA to highlight the advances that computing and data-intensive science are bringing, but on a wide variety of topics, from general relativity to bioinformatics to sociological impact of technologies and so on. So I hope if you're here for the first time, I hope you'll consider following the colloquium series and coming. Today we have a speaker who's going to tell us about advances in genomics and precision medicine, particularly applied to cancer. And I'm really pleased. Had a great discussion last night. He's got a cold, and so we need to have quite a quiet group here, so we'll be able to hear him unless the PA system is loud enough. So bear with us on that. And so Victor Yonganil, who leads the thematic area of computational biomedicine here at NCSA, will do the real introduction.
Thanks very much. So Ed was a warm-up act for me, and I'm the warm-up act for Gustavo. And I don't know if you're bringing somebody along that, no. So thanks much, Gustavo, for coming here. Gustavo is program director of the Translational Systems Biology and Nanobiotechnology group at IBM Research in New York. And he is also now associated with the Mount Sinai Icahn School of Medicine. He was trained as a physicist, initially in Buenos Aires and then at Yale University. And then he became interested in biological problems and wanted to train his analysis skills on biological data. And he's been at IBM for about 20 years now. Correct me if I'm wrong, but it's give or take a few. And probably apart from having done some really exciting research, he's also been an extremely influential person in the biomedical data analysis field by organizing what are known as the DREAM challenges. And the DREAM challenges are, and he will tell us a lot more about those, but are crowdsourcing exercises to try to find the most accurate methods for analyzing complex biological data. And I think the community strongly believes that this is an incredibly important area because a lot of the methods that are being developed and published in this field don't really work very well. And having an open competition that assesses in a neutral way how well different methods work is really a very important contribution to our ability to make sense out of biomedical data. Okay, I'll stop talking here, and Gustavo, again, thanks very much for joining us today.
Thank you, Victor. Okay. Sorry. So, if you read my abstract, what I'm going to talk today doesn't have anything to do with what I say in my abstract because the audience I understand here is a little bit more broad. So I wanted to share with you some of the progress that has happened lately in the area of precision medicine, which I think is fantastic. But I want to convey you the importance of it and put this in perspective, as well as understand the limitations of some of the overhyped claims in the field. I love this phrase. Outside of a dog, a book is a man's best friend. Inside of a dog, it's too dark to read. So DNA has information like a book does. And inside the cell, polymerases and enzymatic reactions read that information all the time. How it's done is fascinating biology and science. But I want to talk today about the incredible advancement that it was to actually learn how to read DNA, and how difficult really it is to do that. I want to give you a little bit of perspective with respect to this sequencing the human genome. So just to, I chose one or two things. Probably you may have other favorite topics of before and after, but imagine the discovery of the Americas in 1492. That was an incredible milestone in itself, and really after that the world was different, somehow got smaller. But what had to happen in order for that discovery to really mean something is that a lot of people had to learn how to use technology to learn, for example, how to map the Americas. Look at this incredible map that Americo Vespucci created. This is supposed how they thought America was in those days. That happened in 1507. They started to have these paintings of the world there. This is incredible technology that is needed to do this. How do you know that the world is like this? They didn't even know how to navigate from east to west. They didn't have a longitude map. Another before and after, if the inventor of the transistor, and I'm saying this here, that the person that co-invented the transistor, Bardeen, along with Shockley and Brattain at Bell Labs, created something that would change the world forever, right? Of course, the transistor allowed computers to exist, and then today's interconnected world through our iPhones and smartphones and so on. But to put it in perspective, this is a floppy disk. I was 10 years old in this year. This thing harbors 100 kilobytes. Today we have these flash drives, and you can put 10 to the 7 things of this sort inside here, just because we miniaturized the transistor in order to put a billion transistors or more in a chip which is very small. To have an idea of this number is the number of times this little sphere that this girl is holding fits in the diameter of the Earth. It's an enormous amount of, a lot of work has to happen for this to happen. And here is chopped, but there were 1,200 transistors that guided the Apollo 11 to the moon. Today, the iPhone 6 has 1.6 billion transistors. And if that computer could bring the Apollo to the moon in 1969, today an iPhone 6 could bring 120 million Apollos to the moon. So imagine the immense advancements. So those advancements really changed the world. I want to submit to you that a similar degree of changing the world before and after is the sequence of the human genome. So, but before we have to say, put this in perspective, these advancements take time, right? This is important for the venture capital, because sometimes they say, I want my return of investment tomorrow. Sometimes that doesn't happen, but that doesn't mean that the investment is not going to pay off eventually. But chromosomes were already seen under a microscope in the 1800s, the mid-1800s. They knew humans have 46 chromosomes, 23 from mom, 23 from dad. But what is inside the chromosomes is something that wasn't very clear until way later on in the mid-1900s, when in 1953 Watson and Crick elucidated the structure of the double helix. Just to give you an idea, each of these chromosomes, you know that if you put together all the bases there, we have 3 billion bases. If you stretch all those 3 billion bases, you get 1 meter, more or less, in length. If you put together all the genomes, all the chromosomes, all the DNA in an adult body, pulled together in a string, which would be very, very thin, but a string that is long, we will get to Pluto, basically. So it's a lot of material that is compacted in these things. And to understand how this is compacted and how it works, it took the ingenuity of these two gentlemen and also the risk takers of this guy, Watson, who didn't know what he was talking about, but he wanted to get, something great done in his life. So the story is that they figured out that there is this double helix, but most important, For me, that I am Argentinian and tango is important. There are two important tango players, Aníbal Troilo and Carlos Gardel. If someone is in tango, will recognize them. AT and CG. So these two guys complementary match each other. And so what was incredible that Watson and Crick realized is that the structure suggests the function. That the fact that one strand contains exactly the same information as the other, but in complementary fashion, implies that if you open this up, you can duplicate that. And they say in their article that it has not escaped our notice. That's how their article ends. And it's a way, they didn't know exactly how this worked, but they said, we have to say it first. And they said it. They said, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So here is where the structure of the double helix really opened up and suggests the function. Well, it took a couple of decades for this gentleman, Sir Fred Sanger, to imagine a way that is efficient to sequence DNA. He got 2 Nobel Prizes. I heard yesterday, one in medicine, one in chemistry. And it was an incredible technological progress that eventually led to the sequencing of the human genome that was used, basically using the Sanger method. Now, to give you a perspective of how, we are creating this map, in 2003, the first genome, which was actually a combination of several people's genomes so that they were not easily identifiable, was first announced, the draft. It took 13 years, estimated 3,000 scientists, and $2.7 billion. In 2007, the second genome was sequenced. That was of a pioneer in genomic sciences called Craig Venter. Some of you might know of him. It took 4 years, 30 scientists, and 100 million. Already you see a couple of orders of magnitude reduction. The third genome was that of James Watson, the Watson and Crick pair, and took 5 months, 30 scientists, and $1.5 million, again, 2 orders of magnitude reduction. The technology now was different from the previous ones, was what is called the next generation technology. The first one of them was called 454, now defunct. Today we have of the order of 250,000 genomes sequenced. 250,000 genomes sequenced. Not about 2 decades ago, less than that. We had just one. Now we have, and we are going to have a lot more very soon. And now the second generation technologies like 454 gave rise to others such as Solid and Illumina, and now we have third generation technologies such as the PacBio, Oxford Nanopores, and Ion Torrent. And probably if Alek Aksemientiev is here, he's going to give us the fourth and last generation technology, which is graphene-based silicon nanopores. People are working very hard on this. The important thing here is to understand that the ability for sequencing the genome fast and cheap increased incredibly over time. in a span of a decade and a half, we went from basically having a $100 million genome to having a $1,000 genome. And when we get to the $100 genome, this is going to become part of medicine. We need a ways to go. But analyzing this is not easy. Sometimes the bottleneck is really trying to make sense of what is in the genome, as we will see in a second. But we have discovered America. We had created the map. Now we have to know what to do with it. Just to make a little branch in the conversation and tell you what I have been working in this field. As Alec knows, I have been trying to design ways of using nanopore technology, very, very narrow pores, hopefully 2 or 3, 4 nanometers in width. Here, there are many cosmologies. You are used to think of the big universe. Now think of the very, very small universe. 2 nanometers, 3 nanometers is just a handful of atoms in length. And when we make a whole of that, basically what you are doing is taking one atom at a time, putting aside and make a whole. So trying to make DNA go through a hole and controlling its passage by interacting with its charges through electric fields is what we try to do at IBM, using the clean room and nanotechnology expertise over there, creating very thin electrodes, 5 nanometers in thickness, separated by dielectrics, and try to make sure that the DNA interacts with this. We run into a wall. I mean, by the way, if you put a voltage across the two sides of the membrane, you get spikes when the DNA goes through, because somehow, in this case, the current goes up, even though sometimes the current goes down. Depends on whether it's bringing ions or whether it's blocking ions. But what is important is, can we control the translocation, and can we measure as we go? And one of the recent papers that came out of my lab and Stuart Lindsay is try to show that you can sequence using solid-state technology that is manufacturable, using nanowires, actually molecular wires, that would distinguish G, A, C, and T simply by measuring the currents and the shape of the current that goes from one electrode to the next through the base that the wires are interacting with. Anyway, this is just a very little window into the depth of what the technology advancements are being done at the nanotechnology and biochemistry. Now, in order to proceed in our story, yes?
Can you just comment on what would be the difference between the $100 genome sequencing that you're talking about getting to and what 23andMe does now for roughly that kind of a price?
23andMe measures not the genome, not the whole genome, which we now know has a lot of information that is essential, but single nucleotide polymorphisms. That means mutations in one base. And so that's much cheaper, much simpler to do. With $500, you can do it. I paid, I think, $100 for mine. I did mine, my son, my daughter, my wife, and I learned that my son and my daughter are my son and my daughter, for example. So that kinds of things you can do, yes? So it's not a whole genome sequence, but single nucleotide polymorphism, and they use a different technology. Let me tell you, and you will know right in a second what kinds of things that technology cannot tell you. Cancer is a disease of the genome. This is something that started to become apparent over the years. But let's see, what was the road that took biologists to conclude that cancer is a disease of the genome? It's not obvious, right? Rembrandt here painted this Bathsheba at her bath. There is a little nodule here that you can see that he very faithfully represented. From looking at these nodules, or these very sometimes uncomfortable for the patient and painful, how do you know that that's a disease of the genome, right? It's not obvious by looking at it. So, but this probably people know, because all of us unfortunately had some experience with cancer through ourselves or our loved ones. Cancer is the second killer disease after heart disease. It kills more people than AIDS, malaria, and tuberculosis combined. Basically, 1 out of 8 deaths in the world is due to cancer. Breast cancer will affect 1 out of 8 women in their lifetimes, and 1 out of 7 men prostate cancer will affect. So if we have 10 people, 10 men in a room, 1 of them probably will have to deal with prostate cancer sometime in his lifetime, same with women. So it's a very serious problem, and as we are living longer, thanks to advances in medicine, will be seeing more and more. Actually, in Africa, this is becoming a problem. The African continent had many problems, but now people are starting to live a little longer, and now cancer is becoming a problem, which before it wasn't. Now, one of the early demonstrations that cancer was created by carcinogens, that something could create cancer, was this experiment by Yamagiwa and Ichikawa. They painted the back of the rabbit's ears with tar, coal tar. There's some, I don't know whether in the back you can see, but for example, they had 15 rabbits and 23 ears. I don't know why they didn't put, I don't know where some ears were lost, but you know. And now, after 70 days, 20 out of these 23 ears developed this folliculoepithelioma cancer, and that's 87%. And the numbers are more or less in the between 60 and 80. If you wait 150 days, 100% of them develop. So that's a very clear, almost no need of statistics, way to say coal tar is a cause of cancer. How? We still don't know at this point. Now, cancer also is caused by tobacco, as we know, 25%, despite the fact that tobacco companies insisted that that was not the case by doing bogus research. 25% of cancer deaths are due to tobacco. And so here is a very interesting plot that shows that if you make a plot of the cigarette smoked per person per year as a function of time, you have a growing curve. 20 years later, this is the curve of lung cancer deaths per 100,000 people. So of course this is only correlative, but it's clear that there is today we know that there is a direct link between the toxins that are in the tobacco smoke and DNA damage. But basically, where I'm going is the following. As our understanding of DNA through genome sequencing and the double helix grew, it was clear that chemicals and radiation introduced DNA damage. And new sequences intercalated in the genome, such as viruses, also led to DNA damage. On the other hand, cancer, at least 15 to 20%, 10 to 20% of cancers are inherited. So families that have cancer have kids that have cancer, depending on what mutations they have, we know today. For example, there is a number of diseases that are known to be cancer. So, and you can look at where the carcinogens are breaking the genome and where the hereditary markers of cancer are, and very often they were similar. So carcinogens create cancer. Carcinogens create cancer because they break DNA, and so that was the elucidation. So during the 1970s, a lot of people started to try to understand where in the genome is the markers of cancer. And so they came to the conclusion that there are two types of cancer genes, the oncogenes and the tumor suppressors. The oncogenes are those that normally have some function, but when they are either broken, their function inhibited, or somehow overexpressed, they can promote growth or inhibit death of the cell growth, and therefore you start to create cancer. And the alterations are very many, like point mutations, like the kinds of things that 23andMe measures, or insertions, amplifications, and translocations, which are things that 23andMe, for example, wouldn't measure. This is an example of a growth factor receptor called HER2, human epidermal growth factor receptor. For women that have amplified their HER2 gene, they die So here you have not amplified, here amplified. If you have more than 5 copies of this gene, because part of the mutations is not that a nucleotide A is changed into a G, it's also that the gene could disappear, or the gene could appear many times, or could be fused with another gene. In this case, HER2 is amplified more than 5. This is how women died after diagnosis of breast cancer with HER2 positive. After 12 months, 40% of, only 40% survived, 60% had died. But if you don't have this amplification, then there is a much higher chance of survival. So here is a clear case of a gene that is an oncogene. While normal cells have about 20,000 HER2 receptors, these are receptors that are at the surface membrane. I will show it in a second, a representation of it. Women with HER2 positive have of the order of 2.5 million. So it's about 1,000 times more, or actually not 1,000 times more, but 100,000 times, 100 times more. Another type of aberration is when a chromosome, for example here, chromosome 9 that harbors this gene called ABL, fuses by recombination with another chromosome. Here is one that is called BCR locus, and they create a fusion. And this fusion is called the BCR-ABL protein, which is an oncogene. When you have BCR fused with ABL, ABL is overexpressed because BCR locus is expressed a lot, and so therefore now that cell has ABL growing a lot, and that is a cause of cancer for chronic myelogenous leukemia or chronic neutrophilic leukemia or acute lymphocytic leukemia. Let's talk about tumor suppressors now. These are the genes that protect us. When they are working okay, typically there is no cancer. But when they are not working okay because they have a mutation or because they have been deleted, these tumor suppressors cannot protect us, and we can have problems in the control of growth, problems in the decision to go to die. The cells have a program called apoptosis, which is basically if their genome is all screwed up or they have to fight with a lot of problems with unfolding of proteins, they themselves shut down the factory and they die. If the cell doesn't know how to go to apoptosis, and p53 is one of those genes that tell the cell go to apoptosis, then this is what happens. This is mice that were exposed to carcinogens. If they have the 2 copies of p53 from mom and dad intact, they didn't die after 500 days. This is the survival, 100% survived. If one of if they are what is called heterozygotes in the mutation of p53, they are plus minus. That means they have one p53 working, the other one is not. Then some of them start to die a little earlier at age 250 days. But when they are p53 minus minus, they don't have any p53. They die much more precipitously. These are called survival curves. And if you look at today, we have a lot of databases with cancer genomes. In ovarian cancer, for example, about 50% of all patients have mutations in p53. p53 is called the guardian of the genome. Is because it has been seen that in about 50% of people that have cancer, either p53 is mutated or a gene that regulates p53 is mutated. One of the works that I have been doing in the past was trying to understand how p53 is regulated in terms of kinetic models, and try to understand whether perturbations such as SNPs in the surrounding area of p53 could cause a disruption of the normal function of p53. And so this is one of the areas in which systems biology are trying to make sense of the genes, not just at the level of I have a mutation or not, but how that mutation quantitatively impacts the normal functioning of the cells. And people are doing a lot of beautiful computational work in trying to create tumor growths, which demands a lot of computational work and tumor growth of many cells that needs a hugely parallel computing capability. So, okay, we know cancer, cancer is a disease of the genome. How can we cure cancer? How people try to cure cancer? So during the 1940s, 50s, and 60s, there was a lot of chemotherapy being developed. And we hear that chemotherapy is great because it can do something for the patient. It's not so good because it really kills the cancer cells, but the other cells that are fragile as well. And so people are trying to have more molecularly targeted approaches that actually surgically modify the growth, for example, of those or the proliferation of cells by making sure that the gene that is responsible in part for proliferation is not active. And so basically they try to repair the molecular defects in cancer by using what is called targeted therapy. So here you have HER2. HER2 is this growth receptor. Cells are not squared, I have to say. So cells are not so three-dimensional. But here you have a very simplistic way of thinking what happens. Cells have to communicate with the outside, have to transmit that information, have to make that information be used in order to transcribe genes that eventually will cope or will do something with the information that came, and also to produce energy to do all these functions and so on. But HER2 is supposed to create growth if there is the right signal outside. Now, for breast cancer, some women have this HER2 amplified, as we said before. In 1988, a company called Genentech in San Francisco, created this monoclonal antibody that bound to HER2, preventing HER2 to receive signals of growth from the outside or from being constitutively active in promoting growth. And so it was approved by the FDA, and of the 30% of women that have HER2 amplified, 30% of breast cancer, women with breast cancer have their HER2 amplified. Of them, 70% benefit from this drug. We'll see that there is more to the story. The same is true for this drug, Gleevec, that was the first rationally designed drug in order to act on this BCR-ABL protein that we discussed before. And this is, again, a progression-free survival curve. People who had the Gleevec survived much longer than people who had other kinds of therapies. And that was the poster child of targeted therapy for many years. It's called Gleevec. And there is a long list now, a long list of targeted therapies. For example, here we have the targets are BCR-ABL, and if before it was Gleevec, there are other BCR-ABL inhibitors like linotinib, for epidermal growth factor, gefitinib, or erlotinib. Then sorafenib for multiple tyrosine kinase receptors and so on. And people were looking at more and more dissecting, if like a decision tree. If a person has this mutation, but has this other mutation and not mutated this, then, like this case, then you have to give this drug, gefitinib. If EGFR, epidermal growth factor receptor, exon 19 has a deletion, or exon 21 has this mutation, and KRAS is wild type, give this one. So this is kind of like a decision. This sounds like great, right? However, in breast cancer, out of the 70% response, 70% relapse, which is case of half full or half empty glass, right? It's great. 70% of the 30% of women that have HER2 positive breast cancer, 70% have something. But unfortunately, only 30% get the gains because the other ones get relapse. The same is true for this BRAF V600E mutation in melanoma. This drug, vemurafenib, has a 50% response, but 90% of them, the cancer come back. And the same with BCR-ABL, 90% survival, which is great, but 30% of them relapse. So what's going on? Why does it happen? What is worse is that there are many more mutations than targets. When you look at the newly sequenced genomes of cancers, 75% of patients harbor no actionable mutations. People that basically go to the doctor says, let's make a biopsy, have the genome sequenced, the cancer genome sequenced, and look at it and say, I don't know what to do with this, right? 75% of them. So there is this problem with interpretation, overinterpretation, and ignorance, and this is, the next 20 years will be about trying to figure out how to really read the genome and understand how to act on them. One of the ways in which we are trying to solve this is by doing combination therapies, because this resistant thing can be for several reasons, but imagine anthropomorphize a cancer cell as a person with two legs, and you cut one leg of the cancer, right? But the cancer can't walk anyway, and eventually it will create some mutations because they like to mutate cancer cells that will create something that looks like another leg and will go. So in order to kill that cancer from being, you have to give a combination that cuts one leg and the other leg. So parallel paths have to be cut so that the cancer doesn't have a chance to continue. There is another problem that cancers are heterogeneous. So there is one group, clonal group within a cancer that is growing with some characteristics, but they are heterogeneous. Some other part of the tumor is growing with some other characteristics. So your drug will be useful for this one, but this one, once this dies, this one starts to expand much more because before this was at least stopped by this one. So there's a complications in all ways. And so some of our work, my work at Mount Sinai, is trying to develop algorithms to imagine how to do, if you know how the monotherapies work, how to do combination therapies to work even better. For example, if you have drug A and B, this is real experiments. Drug A and B kind of not killing, this is viability of a cell culture, not killing it, not killing the cell. In this case, it's a breast cancer cell line. But you put A and B together, and this is what happens. So neither A nor B are killing at this dose. But when you put A and B together, you are killing consistently more. Algorithms to develop these kinds of, to identify these combinations is one of the things that are happening in cancer research. Moving to precision medicine, the concept is to treat the patient, not the disease. It's not that how would we treat breast cancer, HER2 plus breast cancer. It's how do we treat this patient which has not just this disease, but also lifestyle, smoked or didn't smoke, and all these other characteristics. So the idea of precision medicine is to precisely identify parameterize a human, if you will, and understand how to help that human live healthy, or if there is a disease, cure that disease. Medicine could be very imprecise. For example, take Crestor. Many people in America take antidepressants and anti-cholesterol drugs. Crestor helps 1 out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1 out of 20 people that take Crestor really benefit as much as the drug company thinks. So, and this is, one would imagine that a lot more will benefit. But the only requirement from the FDA is that in a statistical test between a group treated with Crestor and one treated with placebo, there is a statistically significant advantage of reduction in the population. So, and that could be that 1 or 2 or 3 have, and that's already enough for. So we need to identify the population that don't respond and try to give that population something else. So precision medicine is an emerging approach for disease prevention and treatment, and the idea is to get not just the individual variations in genes, but also the environmental context and the lifestyle. As you know, last year, President Obama, during his State of the Union, promised $250 million to do what he calls the precision medicine initiative, which is basically try to get 1 million people in the United States to have their genome sequenced and have a database with all their genetics, genomics, and lifestyle characteristics. So it's not the end of the story, and we have heard words of cancer before, but it gives a great push in a direction that is much needed for health. Like Kennedy saying in 1962 that we will put the man in the moon before the end of the decade, and that happened, put a lot of billions of dollars in that, but and we wish we had more money in this field, but I think it's a very good direction to go. So what is necessary? What is necessary is to collect a lot of data, because we still don't understand how to associate these mutations. As we saw, 75% of people we have mutations that we don't understand how to act on. So we need to collect a lot of data. Much of this data will be noisy, but we need to have the data in order to understand where is the signal. And for early cancer detection, we need to monitor what is called liquid biopsies, like circulating DNA in the stream, in the bloodstream, circulating tumor cells, and circulating exosomes. And that's one of the areas of research that I do in my lab. But I won't dwell on this because of lack of time. I want to discuss, however, how we can analyze big data in a way that is a little bit smarter than just one person at a time. And this is by using crowdsourcing. So we know that we have an unprecedented growth of biomedical data, and not just biomedical data, all kinds of data, right? If you are in the biomedical field and genomics field, projects like ENCODE or TCGR or CPTAC, and we just mentioned the precision medicine. So all this data matrix has a great amount of complexity to it. Now, there are new approaches that are needed to analyze this. If the data is so big, you cannot move the data anymore, so you have to have kind of a cloud system and bring the algorithms to the data, and this work is ongoing. Different companies, including IBM, are working trying to facilitate this work and make money out of it. Data sharing is an important, crucial thing as well. We need to be able to share this data, because if I analyze this huge amount of data, it will first take me forever, and second, how do I know that I am the best person to analyze this data set? But there is an issue of culture with respect to the data sharing. Many of you may have seen or not. in one of the top medical journal in the world is called the New England Journal of Medicine. In an editorial written by very prominent physicians, they were talking about data sharing in these terms. A new class of research person will emerge, people who use another group's data for their own ends, possibly stealing from the data gatherers, or to try to disprove what the original investigators had posited. The system will be taken over by what some researchers have characterized as research parasites. Now imagine what this means. It means these people that create data, kudos to them, right? Creating data is difficult. But they are saying, we will not share this data with anybody else. We want to analyze it first, for several reasons. They claim here that it's because they know best. I contend that's not true, but the reality is they want to profit from the fact that they put the work, which is laudable, they want to get the funding that results from the money, from the research that results from that data. They want to publish from it, right? They want to get all the credit. So there is a perverse way in which the system is articulated, in which data sharing is not very much fostered. There is another approach to research which comes to in the analytics, which is create a multidisciplinary team science in which nobody really knows all the aspects of this data, the statistics, the computer science, the acceleration, the domain expertise. One group will not do it. You guys know about this in this NCSA Institute. And the crowdsourcing the question is something that I have a passion for. If I ask a question here, I'm sure among all the people that are here, you know a lot more than just me. So I'm sure that there are many questions that I would not even find in Wikipedia, but some of you will know. But I don't know who is, so I have to ask to everybody. And the next question, it will not be that person that answers, it will be someone else. And so this crowdsourcing idea is basically ask the world how to solve a problem that is important for the world, and there will be answers that will be the right ones, but come from places that you didn't even imagine could come. So crowdsourcing is basically, you have a problem, there is an organization like DREAM, the DREAM challenges that I mentioned before, and that Victor mentioned before, that crowdsources data and a question, the crowd proposes solutions, and the problem is solved, or at least we know the level of solution that we can get. And there are benefits to it. Performance evaluation is one. We are very bad people to analyze our own things. We tend to be very lenient with ourselves, especially when we want to publish our own papers. And so what we will do is, and we wrote a paper about this, we fall in what is called, we call the self-assessment trap, in which we try to find the best metric for our own method, or the best data set for our own method, and then compare and say we are the best, because the referees want to hear that, right? So that's, again, another part of the system that needs to change. So what DREAM, our challenges, and other efforts like that do is to try to give a benchmark in which people can try to solve your problems, or the problems that you give them, but they don't know the answer. They cannot train and test themselves. They train their algorithms, they learn the parameters, but eventually they test their algorithms with the data set that we keep very close, and they don't know. So we have an objective evaluation and a good comparison, apples to apples, between many groups that are trying to check their algorithms work. And the algorithmic developers will know whether their algorithms work or not. That's very important. Oftentimes people think that they are doing very well, but they are just overfitting. We are sampling the space of methods. We are accelerating research. Imagine if we all decide to do try to solve a problem, rather than me alone solving my problem. We will multiply, accelerate the solution times the number of people in the room. And community building, which is basically try to get the dialogue going amongst us, trying to understand the extent to which your idea is better or worse than mine, how they synergize and so on. We don't do that either in the community, right? We tend to compete. We tend to compete for being the first to publish, being the first to get the grant, and so on. And so things have worked well, but in the context of big data, I don't think that's a good idea. So there is something called the wisdom of crowds that probably you guys have heard of, but it's another reason to collaborate, is that Francis Galton, who was a very famous statistician, at some point did the following experiment, motivated for not super nice reasons that we can discuss at some other point. But he said he went to a county fair where people had to estimate the weight of an ox. And he was sure that nobody was going to have any good idea. Actually, he was trying to disprove that democracy works. And the real weight of the ox was 1,198 pounds. The average of these guys were 1,197. So the collective intelligence, if you will, the excesses and deficits in the estimates cancelled, and the mean, if you will, remained. And this, by the way, is a beautiful short paper published in 1907 by Galton in Nature. This is what we are trying to get by doing the DREAM challenges. DREAM stands for Dialogue for Reverse Engineering Assessment and Methods. And we have an active group of people like this one that are willing. Right now we have a user base of about 15,000 people. And they like some challenges, some competitions, not others. But and we converge in a conference that we organize yearly. But they basically get together and talk about why their algorithms work best in one case or the other. So we have organized 40 challenges so far, from network inference to estimation of model parameters, for example. Can we estimate model parameters from data? From, for example, breast cancer prognosis challenge. Given genomics data, can we predict how long a person will live? Or can we predict whether a person with rheumatoid arthritis is going to respond to treatment with anti-TNF-alpha or not from the SNP? And some of you are working on some of these challenges. The somatic mutation calling challenge in in cancer. If I have a germline, that means everything that is not cancer in the person and the cancer, can we say where the mutations are? Which seems easy, but it's not simple. So challenges to benchmark that. We have published this. I'm very proud of how, in the last Nature Methods, we have this cover because there is a paper there reporting on the results of one of the challenges about inferring signaling networks. And this cover is basically, we are like prisoners in Plato's cave trying to look at the shadows of a reality to which we don't have access really. And that's how we feel about network inference. Anyway, the idea of the DREAM challenge is you have a ground truth. You give data about the ground truth, but don't give the ground truth information. For example, who responded to therapy and who didn't. We don't say, but we say what the SNPs are and what is the clinical parameters. We give that data to a lot of teams, and the teams try to predict. And then we can compare. Did you get this patient right? Because I know she was not a responder, but you say it's a responder. How often are you accurately predicting, or how often not? And we can do an objective evaluation. At the same time, we are democratizing this data, giving this data that otherwise would be in the hands of one group to all these people, accelerating research and fostering collaboration. We are running a digital mammography challenge in which there will be a 1 million. We usually don't give so much money, but we got the funding. And the problem is very important, so stay tuned if you are interested in image analysis, how to analyze mammography screening images. But I wanted to discuss something else, the il-Bowen DREAM ALS Progression Challenge, and this is going to be the last 3 or 4 slides in my talk. So, ALS, amyotrophic lateral sclerosis, is a horrible disease, really horrible. If a person gets ALS, very soon they won't be able to walk, they will fall, they cannot tie their shoes, they cannot breathe eventually. And it's always fatal. But some people live a lot, like Stephen Hawking. Some people live very little, like Lou Gehrig. Some people live in between. Our question was, can we predict, based on clinical information, how fast a person will progress in his disease or her disease? So we gave clinical data, and we gave 3 or 4 months for people to work on. And from the clinical data, we asked, can you predict how fast these people will progress? And we got the solutions eventually. There were 2 teams that used Bayesian trees and random forests that did very well. And, but what does it mean to do very well? So let's compare. We took 14 typical progression cases. We gave the clinical data to 12 physicians, experts in ALS, and say, if you have to tell this person how long he will be how fast he's going to progress. Is he going to be a fast progressor or a slow progressor, right? This is the score of progression. Up here is bad, down here is bad prediction. All the doctors were predicting pretty bad in terms of the distance between the prediction and the reality, or the correlation between the prediction and the reality. But the two algorithms were predicting at the between 0.6 and 0.8 correlation, and at the lowest range of the RMS. So that means that these algorithms are learning from the information given in the cohort of people in the clinical trial, and being able to predict with much better accuracy what is going to happen to the person. Definitely much better than the expertise of the doctors, who could be terrific, but they don't have in their way of thinking the information about everything else. They don't have all that statistics so well determined. They don't have a precision with respect to the individuality of the person. And we could figure out also, by looking at the features that people used, we could figure out some of the clinical covariates that were not known to be associated with progression, such as creatinine or creatine kinase or phosphorus in blood. So to wrap up, so the journey that I tried to take you through today was about trying to get some perspective with respect to the sequencing of the human genome. We have witnessed, we are the one or two generations that witness something that will be always different before and after, which is we now have the map of how our biology works. We have the map of, it's not that I say everything is the genome. There is nature and nurture. By the way, Galton invented this concept of nature and nurture. There is environment and so on, but the genome is an essential piece, and we witness that. How do we create the maps that Americo Vespucci created after that, that allows us to go, beyond the targeted therapies that are failing into something which is more of a precision medicine that allows us to use algorithmic technology from learning from evidence that will help us predict when a person is going to get sick, and if that person gets sick, to help that person. And finally, I try to show how crowdsourcing really can get the best out of a data set by really sharing data and making sure that everybody contributes with their expertise to solve problems like cancer or ALS that, unfortunately, the prior says that we will never die. After all, we have never died so far, right? So very unlikely that we die, but we know that we will die. We know that we will get sick at some point. All this thing, not sharing, is shooting in our foot, because only by understanding how health works and disease works, which needs data, because otherwise we are in the blind, completely blinded to that. And all of us analyzing it is that we are going to have a better medicine tomorrow. Thank you.
Thanks much, Gustavo. I'm sure that, we're running a little bit late, but I'm sure you'll be able to take a couple questions.
Yeah.
Thank you. I was wondering, between different types of cancer, one thing that is very common amongst all the cancer patients are the loss of phenotype of the epithelial cells into more mesenchymal characteristics. Going from epithelial phenotype of the cells to mesenchymal, I was wondering if that would be something that would give us a better level of prediction of looking at the cell's phenotype rather than the cell's gene, which is very different in different race and different environment.
Right. The epithelial to mesenchymal transition is supposed to be at the root of metastasis, and metastasis kills 90% 90% of the cancer deaths are due to metastases. So if we can detect that transition that basically loosens up cells, de-differentiates cells to become not part of the mesh of the epithelium in which they live, and start to travel through the bloodstream to colonize other organs and have metastases. So yeah, understanding that is very important. There are drugs that try to prevent that transition, and there are liquid biopsies that try to detect those cancer cells that went awry and started to travel in the bloodstream before they colonize other organs. So that's a very important part of cancer research today, yes.
Thank you. I really enjoyed your talk. My question is, do you have any challenges addressing health disparities? So I'm thinking, for instance, like triple negative cancer that's more aggressive in Black women.
No. We are we don't have cohorts that are so specific as to say, you know, can we detect in a particular genetic background whether we have more or less of that. We're, in a way, the problem that we have in some of our challenges is that even though we have a lot of data, it's not enough. For example, we know in ALS that there is stratification in the disease, and we try to do a challenge for stratifying, and it would be, you know, maybe it's ethnic stratification, maybe it's lifestyle stratification, if it is clinical information. But we always run into the problem that in order to have well-defined groups, you have to have a lot of data. So the short answer is no, but it's something that I think should be done, you know, because it goes in the direction of personalization. No? Triple negative cancer, you don't have HER2 amplification, therefore you don't have Herceptin. You don't have estrogen receptor positive, therefore you cannot use tamoxifen. So it's really a tough one. So it would be very important to personalize to that degree, but we haven't yet.
Okay, any other questions? If not, let's thank Gustavo again.
Thank you.